Neoplasia literally means new growth in medical terminology. It refers to abnormal cell growth that persists even after the initial stimulus is removed. Let's examine how normal cells progress to neoplasia. Unlike normal cell growth, which is tightly controlled and regulated, neoplastic cells exhibit abnormal and excessive growth. To understand neoplasia, we need to contrast normal cell growth with abnormal neoplastic growth. Normal cell growth is controlled and regulated. Cells respond to body signals and stop dividing when appropriate. In contrast, neoplastic cells show uncontrolled proliferation. They ignore normal regulatory signals and continue to grow inappropriately. Neoplasia occurs through a progression from normal cells to cancer. This starts with a mutation, followed by hyperplasia, and eventually cancer develops when cells invade beyond the basement membrane. Neoplasia encompasses both benign and malignant tumors. Benign tumors are contained and don't spread, while malignant tumors can invade surrounding tissues and metastasize to distant sites. In summary, neoplasia represents a spectrum of abnormal cell proliferation disorders, ranging from benign growths to invasive malignant cancers. Neoplastic cells exhibit several key characteristics that differentiate them from normal cells. Let's compare a normal cell with a neoplastic cell to understand these differences. The first key characteristic is uncontrolled proliferation. Unlike normal cells that respond to regulatory signals, neoplastic cells continue dividing inappropriately. Neoplastic cells demonstrate four key characteristics: uncontrolled proliferation, loss of differentiation or anaplasia, abnormal nuclear morphology, and altered cell-to-cell relationships. Under the microscope, pathologists look for specific diagnostic features that help identify neoplastic cells. These cellular features are essential for pathologists to distinguish between normal tissue and neoplasms, guiding both diagnosis and treatment decisions. At the molecular level, neoplasia results from genetic alterations that affect cell growth, division, and death. These genetic alterations fall into three key categories that disrupt normal cellular function. Proto-oncogenes are normal genes that regulate cellular growth and division. When mutated, they become oncogenes that are permanently activated. In normal cells, division is controlled and limited to when the body needs new cells. When oncogenes are activated, they drive excessive cell proliferation, a hallmark of cancer. Tumor suppressor genes normally function as brakes on cell division. They prevent excessive growth and can induce apoptosis when DNA damage is detected. When tumor suppressor genes are inactivated through mutations, such as those caused by UV radiation or smoking, cells lose their ability to control division. DNA repair genes are critical for maintaining genomic integrity. They detect and repair damaged DNA to prevent mutations from being passed to daughter cells. When DNA repair genes are defective, genetic damage accumulates, leading to genomic instability and an increased mutation rate. This diagram shows how DNA damage can either lead to cancer through accumulation of mutations or trigger tumor suppression mechanisms like apoptosis and cellular senescence. Genetic alterations in cancer affect multiple cellular signaling pathways, disrupting normal cell function. This complex network of signaling pathways includes growth factor signaling, cell cycle regulation, apoptosis, metabolism, and DNA damage response. Two critical processes affected by genetic alterations in cancer are the cell cycle and apoptosis. The cell cycle has multiple checkpoints that ensure cells divide properly. Cancer cells often bypass these checkpoints, leading to uncontrolled proliferation. Apoptosis, or programmed cell death, is a complex process that eliminates damaged or abnormal cells. Cancer cells often evade apoptosis, allowing them to survive despite significant genetic damage. To summarize, neoplasia at the molecular level involves multiple genetic alterations that work together to transform normal cells into cancer cells. These alterations create a perfect storm that enables cells to grow uncontrollably, resist death signals, and eventually invade surrounding tissues. Oncogenes are mutated versions of normal genes called proto-oncogenes. In their normal state, proto-oncogenes regulate cell growth and division. When proto-oncogenes undergo mutation, they become oncogenes that drive excessive cell proliferation. Oncogenes can be activated through three main mechanisms. First, point mutations can alter a single amino acid in the protein, causing it to become constitutively active. Second, gene amplification creates multiple copies of the gene, leading to overexpression of the protein. Third, chromosomal rearrangements can create fusion proteins or place genes under control of strong promoters. Let's examine RAS as an example of a critical oncogene. In its normal state, RAS protein acts as a molecular switch that transmits signals from cell surface receptors to the nucleus, leading to controlled cell growth. When RAS is mutated, it becomes locked in the on position, continuously activating downstream pathways and driving uncontrolled cell proliferation. Another important oncogene is HER2/neu, which can undergo gene amplification. HER2/neu amplification is found in approximately 20% of breast cancers. This amplification results in overexpression of growth factor receptors on the cell surface, leading to more aggressive tumor growth. Chromosomal translocations represent another mechanism of oncogene activation, commonly seen in lymphomas. These translocations place oncogenes like MITC, BCL2, and BCL6 under the control of strong promoters, leading to their overexpression. Oncogenes essentially function as accelerators in the cell growth process. In cancer cells, oncogenic mutations keep this accelerator pressed down continuously, leading to uncontrolled growth. Tumor suppressor genes function as the brakes in the cellular machinery. They control cell growth, promote DNA repair, and can trigger cell death when DNA damage is beyond repair. When these genes function properly, they ensure normal cell division. However, mutations can disrupt this process. Two critical tumor suppressor genes are RB and TP53. The RB gene, discovered in retinoblastoma, controls the transition from G1 to S phase in the cell cycle by inhibiting E2F transcription factors. TP53, often called the guardian of the genome, is activated by cellular stress and DNA damage. It can trigger either cell cycle arrest or programmed cell death. The RB protein plays a key role in regulating the cell cycle, particularly the G1 to S phase transition. In its hypophosphorylated state, RB blocks the cell cycle. When phosphorylated by cyclin-dependent kinases, RB releases E2F factors, allowing DNA replication to proceed. The TP53 protein coordinates different cellular responses depending on the severity of DNA damage. Mild damage leads to cell cycle arrest to allow for DNA repair, while severe damage triggers apoptosis to eliminate potentially dangerous cells. The two-hit hypothesis proposed by Alfred Knudsen explains how tumor suppressor genes contribute to cancer development. According to this model, both copies of a tumor suppressor gene must be inactivated for neoplasia to develop. The first hit may be inherited or acquired, while the second hit typically occurs later in life. This explains why individuals with inherited mutations have a higher risk of cancer. When tumor suppressor genes are inactivated through mutation or deletion, cells lose the critical braking mechanisms that prevent uncontrolled growth. This loss of function is a fundamental step in the development of many types of cancer.
Epigenetic changes play a crucial role in neoplasia development alongside genetic mutations. DNA methylation involves adding methyl groups to cytosine bases, typically at CpG islands in gene promoters. In neoplasia, hypermethylation can silence tumor suppressor genes, while hypomethylation can activate oncogenes, both contributing to cancer development. Histones, the proteins around which DNA is wrapped, can undergo various modifications like acetylation, methylation, and phosphorylation. Histone acetylation generally opens chromatin structure, allowing gene expression, while deacetylation compacts chromatin and silences genes. Additionally, chromatin remodelers can physically restructure nucleosome positioning, affecting gene accessibility. Several types of enzymes regulate epigenetic modifications in both normal cells and cancer. DNA methyltransferases, or DNMTs, add methyl groups to DNA and are often overexpressed in cancers. Histone deacetylases, or HDACs, remove acetyl groups from histones and have become important therapeutic targets in cancer treatment. Histone acetyltransferases, or HATs, add acetyl groups to histones and can be dysregulated in cancer. Histone methyltransferases, or HMTs, add methyl groups to histones, and mutations in these enzymes are found in various cancers. Experimental evidence demonstrates the importance of epigenetic changes in neoplasia. This gel electrophoresis shows how gene expression changes when cells are treated with 5-AZA, a demethylating agent. Key findings from epigenetic research show that demethylating agents like 5-AZA can restore expression of silenced genes. HDAC inhibitors can reactivate epigenetically silenced tumor suppressors. Promisingly, combination therapies targeting multiple epigenetic mechanisms have shown effectiveness in treating certain cancers. Several epigenetic drugs have received FDA approval for cancer treatment, including DNMT inhibitors like azacitidine and decitabine, and HDAC inhibitors such as vorinostat and romidepsin. Epigenetic modifications also serve as valuable biomarkers for cancer detection, and methylation patterns can predict treatment response in certain cancer types. Unlike genetic mutations, epigenetic changes are potentially reversible, offering unique therapeutic opportunities in cancer treatment.
Carcinogenesis is the multi-step process by which normal cells transform into cancer cells. The first stage is initiation, where a cell undergoes irreversible genetic damage. During initiation, a cell suffers genetic damage, often caused by exposure to carcinogens. This creates mutations in critical genes controlling cell growth. The second stage is promotion, where initiated cells expand and proliferate. Promotion involves the expansion of these genetically altered cells. Unlike initiation, promotion can be reversible. These initiated cells undergo clonal expansion, dividing to create a population of genetically damaged cells. The final stage is progression, where additional mutations lead to malignancy. During progression, cells acquire additional mutations, leading to increased growth rate and the ability to invade tissues and metastasize. Carcinogenesis typically requires multiple genetic alterations that accumulate over time. This multi-hit model explains why cancer incidence dramatically increases with age, as shown in this graph comparing male and female cancer rates. As tumors progress, cellular processes like autophagy initially decrease during transformation, but later increase to support tumor growth under stress conditions. To summarize, carcinogenesis is a multi-step process involving initiation through irreversible genetic damage, promotion through clonal expansion, and progression with additional mutations leading to malignancy. This process typically requires multiple genetic hits accumulating over many years, explaining why cancer risk increases with age and repeated carcinogen exposure. Environmental carcinogens are external factors that contribute to the development of neoplasia or cancer. These can be divided into three major categories: chemical carcinogens, physical agents, and biological agents. Chemical carcinogens include substances that can damage DNA through direct interaction or through the formation of reactive metabolites. Tobacco smoke is one of the most significant chemical carcinogens, containing over 70 known carcinogenic compounds, including polycyclic aromatic hydrocarbons and nitrosamines. Asbestos fibers can cause mesothelioma and lung cancer through chronic inflammation and direct DNA damage. Industrial chemicals, like benzene and vinyl chloride, are associated with leukemia and liver angiosarcoma, respectively. Air pollution containing particulate matter and carcinogenic compounds contributes to respiratory and other cancers. Physical carcinogens include various forms of radiation that can directly damage DNA structure. Ultraviolet radiation from sunlight causes formation of thymine dimers in DNA, disrupting normal replication and transcription. Ionizing radiation, such as X-rays and gamma rays, can directly break DNA strands, leading to mutations that contribute to various types of cancer. Biological carcinogens include viruses that can integrate into the host genome and disrupt normal cellular functions. Human papillomavirus, or HPV, is associated with cervical cancer and other anogenital cancers. High-risk HPV types produce oncoproteins that interfere with tumor suppressor genes. Hepatitis B and C viruses are major causes of liver cancer worldwide. Chronic infection leads to inflammation, cirrhosis, and eventually hepatocellular carcinoma. Carcinogens can damage DNA through direct and indirect mechanisms. Direct damage includes base modifications, DNA strand breaks, and cross-linking between DNA strands. Indirect damage occurs through inflammation, epigenetic changes, and formation of reactive oxygen species that subsequently damage DNA. These genetic changes can be observed at the cellular level, leading to abnormal cell growth and neoplastic transformation. To summarize, environmental carcinogens from various sources damage DNA either directly or indirectly, initiating the neoplastic process. Individual susceptibility to these carcinogens varies based on genetic factors and other environmental exposures. Chronic inflammation creates an environment conducive to neoplastic development through several mechanisms. There are three key mechanisms through which inflammation contributes to cancer development. First, inflammation leads to DNA damage through the generation of reactive oxygen species. Second, chronic inflammation drives increased cell proliferation and survival, creating more opportunities for mutations to occur. Third, inflammation alters the immune response, which can suppress the body's natural ability to eliminate abnormal cells. Let's explore two important clinical examples of how chronic inflammation leads to cancer. Inflammatory bowel disease, particularly ulcerative colitis, significantly increases the risk of colorectal cancer. Chronic inflammation in the colon leads to a progressive series of genetic mutations, including in key genes like APC, KRAS, and TP53, ultimately resulting in colorectal cancer. Another important example is chronic hepatitis, which is a major risk factor for liver cancer. Chronic hepatitis, whether caused by viral infections or other factors, leads to persistent inflammation, cirrhosis, and eventually liver cancer. Given the strong connection between inflammation and cancer, anti-inflammatory drugs may have significant cancer preventive effects. Non-steroidal anti-inflammatory drugs, such as aspirin, inhibit cyclooxygenase enzymes and reduce prostaglandin production. Studies have shown that regular use of these medications may reduce the risk of certain cancers, particularly colorectal cancer. In summary, the connection between inflammation and cancer explains why chronic inflammatory conditions increase cancer risk, why anti-inflammatory approaches may help prevent cancer, and why controlling inflammation represents an important therapeutic target in cancer management. Clonality of neoplasms refers to the origin of cancer cells from a single progenitor cell. Most neoplasms are monoclonal, meaning they arise from a single altered cell that undergoes uncontrolled proliferation. This initial altered cell contains genetic mutations that enable it to bypass normal cell cycle controls. As this cell divides, it produces a population of identical cells, all carrying the same initial genetic alterations. One key piece of evidence for the monoclonal origin of cancer comes from X chromosome inactivation studies. In females, one of the two X chromosomes is randomly inactivated in each cell during embryonic development. In normal tissues, this results in a mixed pattern of maternal and paternal X chromosome inactivation. However, in neoplastic tissues, all cells show the same pattern of X chromosome inactivation, indicating they all derived from a single precursor cell. Modern genetic analysis techniques provide further evidence for the monoclonality of neoplasms. Genetic sequencing reveals specific mutations that are consistently present across all cells within a tumor, confirming they originated from a single mutated cell. Although neoplasms begin from a single cell, they evolve over time as additional mutations accumulate. This leads to the development of subclones within the tumor, each with distinct genetic profiles and potentially different levels of aggressiveness. To summarize, most neoplasms are monoclonal in origin, arising from a single altered cell. This has been demonstrated through X chromosome inactivation studies and genetic marker analysis. As the neoplasm progresses, additional genetic alterations lead to the development of subclones with varying degrees of aggressiveness, contributing to tumor heterogeneity. Grading and staging are two systematic approaches used to assess cancer severity and guide treatment decisions. Tumor grading assesses the degree of differentiation and cellular atypia in neoplasms. It's based on how closely cancer cells resemble normal cells under a microscope. As shown in this image, grade 1 tumors have well-differentiated cells that closely resemble normal tissue. Grade 2 shows moderate differentiation, while grade 3 represents poorly differentiated cells with marked variation in size, shape, and arrangement. Higher grade tumors with poorly differentiated cells just generally indicate more aggressive cancer with a worse prognosis. Tumor staging, on the other hand, evaluates the extent of cancer spread throughout the body. It considers tumor size, lymph node involvement, and the presence of metastasis. The TNM system is the most widely used staging system. T describes the size and extent of the main tumor. N indicates whether cancer has spread to nearby lymph nodes, and M shows if cancer has metastasized to other parts of the body. Let's break down the TNM system in more detail. The T component describes the primary tumor's size and extent. As the T number increases from T1 to T4, it indicates a larger tumor or more extensive invasion. The N component describes the spread to regional lymph nodes. The progression from N0 to N3 indicates increasing lymph node involvement. Finally, the M component describes whether cancer has spread to distant sites. M0 means no distant metastasis, while M1 indicates that distant metastasis is present. This illustration shows how cancer progresses from a localized tumor to an invasive cancer with metastasis. As cancer advances through these stages, the TNM classification changes, reflecting the increasing severity. Both grading and staging systems have important clinical applications. They help determine prognosis, guide treatment decisions, and facilitate communication between healthcare providers. Large healthcare databases, like the SEER program shown here, collect staging and outcome data to continually refine our understanding of cancer prognosis and treatment effectiveness. In summary, grading and staging together provide a comprehensive assessment of cancer severity and guide personalized treatment approaches for each patient. Tumor markers are critical biological indicators in the field of oncology. Tumor markers are substances produced by cancer cells or by the body in response to the presence of cancer. There are several types of tumor markers that clinicians use in cancer management. Protein markers include prostate-specific antigen, or PSA, for prostate cancer, and CA-125 for ovarian cancer. Hormone markers include calcitonin for medullary thyroid carcinoma and human chorionic gonadotropin, or HCG, for testicular and gestational trophoblastic cancers. Enzymatic markers, such as alkaline phosphatase, can indicate bone or liver cancer involvement. Genetic markers like BRCA mutations for breast and ovarian cancer risk assessment, and HER2 expression for breast cancer treatment decisions. Let's examine how tumor markers are utilized in clinical practice. Tumor markers can be used for screening high-risk populations, though their sensitivity and specificity often limit this application. While rarely diagnostic on their own, tumor markers provide supporting evidence when combined with other diagnostic methods, such as histopathology. One of the most valuable applications is monitoring treatment response and disease progression. Tumor markers excel at detecting cancer recurrence, often before imaging studies can detect physical changes. Despite their utility, tumor markers have important limitations that clinicians must consider. Understanding the appropriate use and limitations of tumor markers is essential for optimal patient management in oncology. Angiogenesis is the process of forming new blood vessels from existing ones. Neoplasms can only grow to about 1 to 2 millimeters in size without their own blood supply. Beyond this size, diffusion alone cannot provide sufficient oxygen and nutrients. The transition from a small avascular tumor to a larger vascularized one is known as the angiogenic switch. The difference between vascular and avascular tissue is crucial for understanding tumor growth limitations. Vascular tissue has blood vessels that provide efficient nutrient and oxygen delivery, while avascular tissue must rely solely on diffusion. Neoplastic cells secrete angiogenic factors to stimulate the formation of new blood vessels. The most important of these is VEGF, vascular endothelial growth factor, which plays a critical role in signaling blood vessel growth. VEF binds to receptors on endothelial cells, triggering multiple signaling pathways that promote cell survival, proliferation, and migration, all necessary for new blood vessel formation. Let's look at how tumor angiogenesis actually occurs. Tumor cells secrete VEF, which diffuses through the tissue and reaches nearby blood vessels. This triggers the sprouting of new vessels that grow toward the tumor, following the VEF gradient. Once connected to the tumor, these new vessels provide essential oxygen and nutrients, enabling rapid growth. They also create potential pathways for tumor cells to enter the bloodstream and metastasize. Angiogenesis doesn't just support tumor growth, it's also critical for metastasis. The new blood vessels formed during tumor angiogenesis have several characteristics that make them ideal for cancer spread. They're often leaky and have incomplete basement membranes, making it easier for tumor cells to enter the circulation and spread to distant sites. Tumor blood vessels are also abnormal in structure and function. Unlike normal blood vessels, they're often disorganized, dilated, and can have excessive branching or dead ends. This contributes to uneven blood flow within tumors and can affect how treatments reach cancer cells. To summarize what we've learned about angiogenesis in neoplasia, tumors cannot grow beyond 1 to 2 millimeters without new blood vessels. VEF is the primary angiogenic factor that drives this process. The new vessels provide oxygen and nutrients, are structurally abnormal, and create pathways for metastasis. Perineoplastic syndromes are medical conditions triggered by a cancer in the body, but they're not caused by the tumor's physical presence. These syndromes occur when tumors secrete bioactive substances like hormones, cytokines, antibodies, and peptides that affect distant organs. Perineoplastic syndromes can be categorized into several major types. These include endocrine syndromes from hormone production, neurological syndromes from antibodies attacking neural tissue, hematological syndromes affecting blood cells, and metabolic syndromes disrupting normal body processes. Cushing's syndrome is an important endocrine perineoplastic syndrome. It's most commonly associated with small cell lung cancer and caused by tumor cells producing ACTH, a hormone that normally comes from the pituitary gland. Hypercalcemia is a common metabolic perineoplastic syndrome, especially in patients with lung cancer, breast cancer, and multiple myeloma. It occurs when tumors produce PTHrP, which mimics parathyroid hormone and disrupts normal calcium regulation. Neurological perineoplastic syndromes occur when the immune system targets nervous system components after being triggered by similar antigens on tumor cells. These disorders can affect various parts of the nervous system and often present before cancer diagnosis. Hematological perineoplastic syndromes affect blood cell production. These include conditions like eosinophilia, granulocytosis, and pure red cell aplasia. They're typically caused by tumors producing cytokines that interfere with normal blood cell development. Perineoplastic syndromes have significant clinical importance. They're often the first sign of cancer, sometimes appearing before the tumor is large enough to detect through standard testing. When treatment is effective, these syndromes typically resolve. Their reappearance may signal cancer recurrence, making them valuable markers for monitoring treatment success. Hereditary neoplastic syndromes are cancer predisposition conditions caused by inherited genetic mutations. These syndromes share several key characteristics that distinguish them from sporadic cancer cases. Familial adenomatous polyposis, or FAP, is caused by mutations in the APC tumor suppressor gene. Patients develop hundreds to thousands of polyps throughout the colon, as shown in this surgical specimen. Hereditary breast and ovarian cancer syndrome results from mutations in BRCA1 or BARA2 genes. This inheritance diagram shows how the mutation can be passed from parent to child with a 50% probability. Lynch syndrome, also known as hereditary non-polyposis colorectal cancer, is caused by mutations in DNA mismatch repair genes. These patients have fewer polyps than FAP, but still carry a high risk of developing colorectal and other cancers. Most hereditary cancer syndromes follow an autosomal dominant inheritance pattern. This family pedigree shows how cancer can appear across multiple generations with earlier age of onset and multiple affected family members. Identifying hereditary cancer syndromes is crucial for implementing targeted screening protocols for patients and their family members. Diagnosing neoplasia requires multiple complementary approaches to ensure accurate detection and characterization. Clinical examination begins with careful visual inspection and palpation. Two-step algorithms help clinicians evaluate suspicious skin lesions. This includes thorough assessment of symptoms and risk factors. Imaging studies provide critical visualization of internal structures. PET scans, shown here, detect metabolic activity in tumor cells by tracking radioactive glucose uptake. Other modalities include X-rays, CT scans for detailed cross-sections, and MRI for excellent soft tissue contrast. Laboratory tests analyze blood and other bodily fluids for signs of cancer. These include complete blood counts, specific tumor markers like PSA for prostate cancer or CA-125 for ovarian cancer, liver and kidney function tests, and hormone levels when appropriate. Pathological examination remains the gold standard for definitive cancer diagnosis. Tissue samples obtained through various biopsy procedures are examined under a microscope by pathologists. Histopathology analyzes cell structure and organization, while immunohistochemistry identifies specific protein markers characteristic of certain cancer types. Molecular diagnostics identify specific genetic alterations in tumor cells. Techniques include PCR to amplify DNA sequences, next-generation sequencing to map the entire cancer genome, and fluorescence in situ hybridization to detect chromosomal abnormalities. These methods identify actionable mutations that can be targeted with specific therapies. Liquid biopsies represent a revolutionary advance in cancer diagnostics. These non-invasive blood tests detect circulating tumor DNA shed by cancer cells. They can monitor treatment response in real time, identify emerging resistance mutations, and detect minimal residual disease after treatment, potentially allowing earlier intervention for recurrence. Modern cancer diagnosis relies on an integrated approach. Different diagnostic modalities complement each other to provide a comprehensive assessment. This multimodal strategy improves diagnostic accuracy and helps guide optimal treatment selection for each patient.
If you enjoyed this video and would like to practice your understanding, go to matheasy.com. There, you'll find this lesson outlined and divided, with a short quiz to see how well you really understood it. And if you're hungry for more, we've got piles of other topics for you to explore. Start learning easier. Visit MathEasy today.